Moving on to chapter 3, we'll look at the level of organization at the cellular level. And so this is where we get to look at the structure and function of cells, which are the basic building blocks of all plant and animal life. And cells were first introduced, or I should say discovered, when the microscope was invented and developed. And one particular researcher, Robert Hooke, looked at quark and he discovered that there are tiny little subdivisions that were reminiscent to him of rooms in a monastery or in a prison, and hence the name cell was coined. So cells are the building blocks of all plants and animals. And as researchers began to study them and people took an interest in what was going on at this very small level that was before invisible, before when all researchers could do was look with the naked eye, once they were able to see on the smaller level, they got quite interested in these little tiny things and how they worked. And they discovered that all cells come from the division of pre-existing cells. And then they figured out that cells were the smallest units that perform vital physiological functions. And in fact, there are some organisms that are simply a single cell. So single cell organisms exist as one cell that is able to carry out all the functions that it needs to maintain homeostasis and allow that little single cell organism to live. Interestingly, this is carried through in larger organisms as well. Each cell must be able to maintain homeostasis for itself. Otherwise, it will die. So each cell maintains homeostasis at the cellular level, as well as performs the function that enables it to work in the larger tissue and organ system in which it is found to have this integrated homeostasis of the entire body. There are two types of cells. We'll be looking mainly at somatic cells, and somatic cells are the cells that comprise the body. For example, your liver, your kidneys, your brain, all of these things. The sex cells are very specific to reproduction of the organism. So you have two types of sex cells. The male carry sperm, the female carry oocytes. And when these two types of cells combine, they can eventually become a completely new individual. And we'll study them a little bit when we get to the chapter on reproduction, but for the most part, we're going to be looking at somatic cells, or cells that make up the parts of the body and the organs of the body. So here is a generic cell, and this is on page 66 and 67 in your spotlight in your book. This particular cell is kind of a generic cell in that it shows all the types of organelles and components that we see in a typical cell. However, it has some organelles that you may see in some cells, but not others, because in the body we have very many specialized tissues and organs, and cells within those systems may vary depending on their function. So for example, this cell looks like an intestinal cell because it has microvilli, and microvilli are often associated with intestinal cells, and we'll see why when we go into the digestive system. And we'll talk a little bit about the function of these things a little bit later in this chapter. But this particular cell, our model cell here, has most of the organelles and components that we will see in typical cells within the body. Now, these may vary depending on the cell function and where it's found. But this is a good model because it shows most of the systems or most of the components that we're likely to encounter in any of the cells that we study. Now, suffice it to say that an intestinal cell will look quite different from a muscle cell, which will look quite different from a blood cell. And we'll be able to study each of these individual types of cells later when we get into those particular systems. But for now, we want to look at an overview of, first of all, what is a cell and how does it work? Now, a cell, as we said, is the smallest unit that's able to maintain all vital physiological functions, and it has to maintain homeostasis for itself. But we can think of a cell as kind of like a little city. So we've got this border here called the plasma membrane, and this surrounds the cell and identifies, well, this is a cell. This is the border. It's also known as the plasma lemma. So everything inside of this thing is part of the cell. Everything outside of this thing is going to be part of the extracellular fluid. Now, inside the cell, we have various components, which we call organelles. And the organelles perform very specific functions. And not at all unlike a city, we have different structures here that are very similar to what you might find in a city. For example, we have this structure here called the endoplasmic reticulum. This is very much like a factory and a warehouse complex. We have this structure here called the Golgi apparatus, which is very, very similar to a shipping hub or a FedEx hub or what have you, where proteins and other products that the cell makes will be prepared and packaged for final shipping to either within the cell or outside of the cell. So we have other little organelles here, like this mitochondrion, which could be considered kind of like a refinery that's making energy for the cell. So this cell is entirely self-sufficient. It can support itself, but it also has functions that it is able to perform that will affect other cells. So in a city, let's just make the example of High Point, for example. One of the big exports of High Point, North Carolina, was furniture and textiles. So not only is High Point a city that can sustain itself and has everything it needs to keep the functions of the city going and so that the city can continue operating as a city, it also exports things. It exports furniture and hosiery to other cities across the United States and even across the world. So in that way, you can see some cells in the body being like that. They will produce products that will be exported to cells and tissues all over the body. And so in this way, you can make the analogy that the cell has this function very much like a city, and we have all of these little things inside it that perform functions that are very similar to things you might find within a city. Now, what we have on the inside of the cell, this entire stuff inside, is called the cytoplasm. Everything inside the cell is called the cytoplasm, and everything outside the cell is called the extracellular fluid. Now, the cytoplasm consists of a watery fluid called cytosol and all the things that are suspended within it, like these organelles and other things. Now, the cytosol also has ions and soluble proteins in it. And the cytosol, though its primary component is water, it is quite different from the extracellular fluid, which primary component is also water. However, what is dissolved in the water inside versus outside the cell is quite different. And that's one reason that the plasma membrane, also known as the plasma lemma, is so important because it can regulate what gets in and outside of the cell. It regulates what exports the cell can send out and what imports it can receive. And so it's very important. It is the interface of the cell with the rest of the environment. It's the first part of the cell that senses changes in the external environment. So then within this cytoplasm, we have all of these different organelles. I've already gone through a few, but let's start with this one in the center, the nucleus. The nucleus contains the genetic repository, so it contains your DNA. And here is where the instruction set for every protein and everything the cell will do is contained. So every protein that's manufactured by the body, the instruction set for making that protein resides in the DNA, which resides in the cell nucleus. Now, as we go outside the cell nucleus, we can see all these organelles that are important for different things in the cell. We've already mentioned this one, the endoplasmic reticulum. It's kind of like a little factory and warehouse because what does it do? It produces carbohydrates, lipids, and proteins. And you can see there are two types of endoplasmic reticulum. We'll go into them in much greater detail later on, but you have the smooth type, which produces carbohydrates and lipids, and you have the rough type, which produces proteins. They are distinguishable by these little blue studs in this picture. The rough endoplasmic reticulum is studded with these little blue, small, round things. And the smooth endoplasmic reticulum completely lacks these things. Now, these things are called ribosomes, and ribosomes is where proteins are assembled. So you have fixed ribosomes that are stuck on the rough endoplasmic reticulum, and this is where protein synthesis will take place, and then the protein will be released into the endoplasmic reticulum where further modifications will be made. We'll look at that in a little bit. Then we have these floating ribosomes that are just floating around in the cytoplasm. And sometimes some proteins will be assembled at these little ribosomes and released directly into the cytoplasm. And these ribosomes are basically little tiny protein assembly factories. Here we have a mitochondrion. As I said earlier, this is kind of like your cell refinery. This is where ATP is made. As we looked in the last chapter, ATP is a way to store high energy bond, phosphate bond. On adenosine diphosphate, you add a third phosphate to get adenosine triphosphate, abbreviated as ATP, and that becomes a major energy source for the cell. You have other important components of the cell. For example, you have your kind of like your garbage collectors here, your lysosomes or your recycling plants. They'll take old and damaged organelles and they will recycle the proteins and the components of the organelle and basically digest things in the cell that you don't want to be in the cell. Sometimes it'll digest by bacteria and other things that get in there. You have other little recycling plants, some called peroxisomes, and these break down fatty acids. You have an immense scaffolding network made of different kinds of filaments called microfilaments. We have microtubules. We have something called intermediate filaments, and these are kind of like scaffolding for this cell. In the middle of the cell, near the nucleus, you have something called the centrosome, which contains two centrioles. We'll look at those in a little bit. These are made of microtubules, and these basically erect and deconstruct the scaffolding of the cell. And so this just basically gives you an overview of what the cell looks like. We have also microvilli here, as we said earlier. Now, what do these do? They're little finger-like projections, and as you can see, they increase the surface area of the plasma membrane. So in an area like the intestines where you need to absorb things, this increases the surface area for absorption. You'll also see something called cilia, which are not on the cell, but you'll see them in the cells of the bronchi, of the lungs, and the nasal passages. Cilia have like little beating, they're almost like hairs, they're hair-like projections, and they have specific functions in moving things along passageways outside the cell. And we'll look at all of these things as we study the cell in greater detail. So let's first look at the plasma membrane, because this is the border between the inside and the outside of the cell. The plasma membrane, as we just said, separates the intracellular fluid, which is called the cytoplasm, from the extracellular fluid. And the cytoplasm, as we said, contains all the structures called organelles and the cytosol, which is the liquid in which they're suspended. Now, as I've said earlier, the cytosol is primarily water. It has some proteins suspended in it, and it has some ions suspended in it, whereas the extracellular fluid is also watery, mostly water, but the ions that are in it are quite different, and it doesn't have anywhere near the amount of proteins in it that the cytoplasm has in it. So here it is, and we can look, it looks like it has some complexity to it, but it really isn't. At the core of the thing, we have what we call the phospholipid bilayer. Look at this in a little bit of detail, but you probably remember when we talked about how certain molecules can like water or not like water. In the case of a phospholipid, it has a hydrophilic head that likes water and hydrophobic tails that don't like water. So what these things do is they assemble themselves to make the plasma membrane in what we call a bilayer, because the heads of the phospholipids will orient toward the watery, in this case, the external part will orient toward the extracellular fluid, and the internal part, the hydrophilic heads, will orient toward the watery cytoplasm. And then the hydrophobic tails will aggregate together on the inside. So this is called the phospholipid bilayer because you have two layers of phospholipids that are facing with their heads away from each other. We also mentioned earlier how cholesterol is an important molecule for stiffening this membrane and giving it some rigidity so it can hold its shape. Now these are cholesterol molecules right here. We also have, in addition to phospholipids, we have some of these which are glycolipids. And these, instead of having a phosphate group with some organic component on top of it. We have instead a carbohydrate. In this case, it looks like a fairly complicated sugar here. In addition to these things, we have proteins that are integral to the phospholipid bilayer. That is to say that they reside within this membrane. And if you were to take these proteins out, you would actually damage the membrane. And these proteins have all kinds of different functions. These are called integral proteins. We also have some proteins that are called peripheral proteins. These proteins can kind of detach and take off somewhere and go inside the cell and do stuff and then come back and reattach to the plasma membrane. These are proteins that if you took them away or dissociated them from the plasma membrane, it wouldn't do any damage to the plasma membrane, whereas the integral proteins are embedded in the plasma membrane in such a way that you couldn't really take them out. Now, what do these integral proteins do? Well, we've already discussed the fact that we have this phospholipid bilayer. Well, things can't just easily go through this phospholipid bilayer because things that are not fat soluble can't go through them. They don't like, the hydrophobic tails don't like water. Ions really aren't going to pass through this thing. So there has to be a way for proteins and ions and other things to get into the inside of the cell. And this can be tightly regulated, but we have these proteins that can act as channels that can let certain things through. These glycoproteins, or glycolipids in this case, that are also part of the cell membrane are signals. They're kind of like little flags that stick up and say, hey, I'm a regular cell. You know, I'm normal. Don't attack me. So the immune system can recognize these things as markers or flags, if you will, of normal cells that belong within inside the body. And the whole group of these things, when you have these glycoproteins and glycolipids, the carbohydrate part that sticks up on the external part of the cell, all of it together is called the glycocalyx, and it forms kind of this sticky surface on the cell. Now, going back to the integral proteins here, these have a variety of forms and functions. We'll look at them in a little bit, but think of them basically as your gatekeepers, your channels. They act like little tunnels and little gates where molecules can get in and out of the cell. A lot of times they're important for helping to attach. Integral proteins are important for helping to attach the plasma membrane to underlying scaffolding, like your microfilaments that we discussed earlier. So this is kind of an overview of what the plasma membrane looks like. Obviously, some of the functions are physical isolation to keep things out of the cell that you don't want in there. Regulation of exchange with the environment. This is how nutrients and ions enter. And also how things that you don't want in the cell, like waste products, are released or sent out. It has that sensitivity to the environment, so it can sense changes in the extracellular fluid composition or pH changes or things like that, and then it can help the cell react appropriately. And it has chemical signals. We already saw the glycocalyx, those carbohydrate portions of the integral proteins and the glycolipids. Those things act as flags that alert the immune system that this is a normal cell and you don't want to attack it. We also see that the plasma membrane has structural support functions. So some of these integral proteins help bind the phospholipid bilayer to the internal scaffolding of the cell, and also you have anchors between cells. And we'll look at some of those a little bit later on, but you have ways that cells will stick together, and they have proteins that and other molecules that they sort of pass between one another as like little fasteners, and we'll see that a little bit later on. All right, here it is again. Here you can just sort of look over. Here's a channel where things can get through. Here's a channel that we call a gated channel. You can see it has kind of a gate, and this gate can be either opened or closed. Right now it's in the closed position, so nothing's going to get through. You see these little indentions here. This is where something can come in a molecule can come in, or an ion, and bond to this thing, which will then open the gate. And we'll look at that in a little bit as well. So to remind you a little bit about the phospholipid bilayer, to remind you the structure of a phospholipid, you probably remember from the previous chapter that you have basically a diglyceride. So what is that? You've got your glycerol molecule right here. That's this 3-carbon sugar. And then attached to the glycerol molecule, you have one fatty acid right here. Remember, it links through its carboxyl group at the head to the hydroxyl group that's on the glycerol molecule. And then you have a second, in this case, a second fatty acid. And notice this one has a double bond. So it is now a mono unsaturated fatty acid. And it has this kink in it. That's very important because the double bond introduces this kind of kink that gives it this sideways kind of dogleg appearance. And then we have this phosphate group right here. And then we have some other molecule that's attached, or component, I should say, molecular component that is attached to the phosphate group. Now, this is important because the phosphate group, with all its oxygens, and here we have a nitrogen, these are all hydrophilic. They like water. This part is very hydrophobic. So notice how this thing orients. Here's the head of the thing. Well, it's abbreviated. This whole part is abbreviated by this green sphere in our little model, whereas the purple part represents these little tails here, the fatty acids. Notice how the fatty acids are all on the inside, and the hydrophilic heads are facing away from each other, one oriented to the extracellular fluid and one oriented to the cytoplasm. This is just a reminder of what a phospholipid is and how the phospholipids would behave in the cytoplasm, the cell membrane. All right, so remembering how these phospholipids and glycolipids behave if you just throw them in water, what do? They form these micelles, remember? So you have your hydrophilic heads oriented toward water and the hydrophobic tails oriented toward one another. Whereas in the cell, because you have both an intracellular fluid that's watery and an extracellular fluid that's watery, what these things tend to do is make this bilayer where the hydrophobic tails are oriented towards themselves and the hydrophilic heads are oriented away. Each in the external part of the cell, they're oriented toward the extracellular fluid, and in the internal part of the cell, they're oriented into the cytoplasm. So we talked about what the phospholipid bilayer is and why it works the way it does. But this, because of the fact that it's got these hydrophobic tails, it does not let ions in water pass. So that's why we have some of these integral proteins, like these here, that act as channels to let things in or to transport things out. So we've got these integral proteins that we discussed that act in this way. And we also have some peripheral proteins that we already discussed that can detach, like this one here, and do something out in the cell and then come back. When we look at second messenger systems later on, we'll see why this becomes important. Because sometimes things will happen outside the cell and will signal something by binding with something on the surface of the cell. And some of these peripheral proteins will then be induced to detach and go off and do things in the cell, and then come back when they're done and reassociate themselves with the internal part of the cell membrane. So we have both integral membranes and peripheral proteins. Membrane proteins have multiple functions, and we already looked at some of them. Some of them are channels that let water and solutes through the membrane. Some of these channels are kind of gated, which we saw, and you have to open and close them to let things in and out. Some are more like tunnels. There are things called aquifers that basically let water right on through. But water can't pass through the membrane itself, so it has to have a special channel that lets it through. So one of the things, obviously, we saw is we have anchoring proteins. These are proteins that anchor that cell membrane to the underlying microfilaments and microtubules and intermediate filaments. And we'll see this especially when we look at microvilli. We'll see how they're anchored to something called a terminal web, which is a bunch of these structures these scaffolding structures that help the cell keep its shape. Then we have our recognition proteins. We saw some of those in the glycoproteins, and there are others that basically act as flags. And these things will label a cell as normal, or if for some reason something should go wrong and a cell should be attacked by bacteria and can't fight off the infection, then it might send up a signal that says, hey, we need help over here, and the immune system will come in and sometimes even destroy the entire cell rather than let the infection spread. Then we have enzymes, and we have enzymes that some of which are actually attached to the plasma membrane, and these will catalyze reactions. Just as they will, we have some enzymes that are floating around within the cell as well. And as you recall, an enzyme, biological enzymes, are proteins that help speed up chemical reactions that otherwise wouldn't occur in the body or would occur so slowly that they wouldn't do much good in terms of any physiological process. Then we have our receptor proteins. We already looked at some of these, like some of those gated channels. For example, this one. Here's a gated channel, and it has a little place for something to bind to it. What binds to it is called a ligand. And a ligand can be any kind of molecule, such as a hormone, or an ion, such as calcium. And what happens is when a ligand comes and binds to that little pocket there, right here, then the gate will open and things can pass through. Now, these channels are pretty specific for particular molecules. In some cases, you'll have carrier proteins. Instead of having channels that are either tunnels or gated channels, you'll have carrier proteins that act as like chaperones, and they'll be on one side or revolving doors. They'll pick up molecules or ions on one side of the extracellular fluid, let's say, and then bring them into the cell. And they work in reverse as well. They're like little revolving doors. That's a good way to think of them. So here's a ligand-gated channel. Here we see a molecule coming, and you can see that it's closed. This is a gated channel, so it's a gate and it's closed. The ligand, that's this thing right here, comes and lands in this little pocket that's specific to it. So it's kind of like a little key that opens the gate. And once the ligand has landed in the pocket and bound to the pocket, that changes the conformation of the protein and opens the gate, so to speak. Ligand-gated channels we will see over and over again. And these are very important, especially when we get to the chapter on muscle and nerves. So the other thing we looked at was we already looked at proteoglycans, glycoproteins. These are proteins with carbohydrate components, and lipids, in this case of glycolipids, with carbohydrate components. And these are the ones, these things sticking outside of the cell membrane. These form a sticky, what we call sugar coat, called the glycocalyx, and it makes it sticky, and a lot of times this is important for cell mobility, or in the case where you have other cells that are just fixed in an organ, sometimes it will attract, this glycocalyx will help attract and slow down things within the blood. This is especially important in immune function, and so it acts as kind of a substrate for things to stick to. So it has a lot of functions. For example, it lubricates and protects the cells as well, so that things can sort of slide over it sometimes pretty easily. And it also helps in anchoring and locomotion. There is specificity in binding. For example, some of those little carbohydrates sticking up act as receptors, and some of them act as little flags that alert the immune system as to the status of the cells. Everything's okay here, we're all good, this is a normal cell, or something's wrong here, we need help. And here is an example of a glycolipid. Here is a glycoprotein, and what makes it so is it has this little carbohydrate part of it sticking up, and all of these together make the glycocalyx. So now that we have examined the plasma membrane itself, we can look at what's inside the cytoplasm, which is contained within the cellular membrane. The cytoplasm is everything inside the cell, but it doesn't include what's inside the nucleus. The nucleus itself we'll consider as a separate section. It's an organelle, but it's very specific as what goes on in there. As we've said, that is the repository for your genetic material where your DNA is stored. So inside the cytoplasm, we can divide that up into the fluid part, which is the cytosol. This has dissolved materials in it. And it's important to note that inside the cell, the cytoplasm has a high concentration of potassium. It has very low sodium concentration. In the extracellular fluid, quite the reverse is true. You have in the extracellular fluid higher concentrations of sodium and lower concentrations of potassium. We have more protein dissolved in the cytosol than you have dissolved in extracellular fluid outside the cell. You also have a higher carbohydrate within the cytosol. Now, the organelles are the structures that we see, and both of these things are contained within the cytoplasm. So you can think of the cytoplasm as a suspension with all these things in it, the cytosol being the fluid part. The organelles, as we've already sort of walked through in our first look at our spotlight on page 66, 67, the organelles are structures with our very specific functions. We've touched on these briefly, and now we can go into a little bit more detail about them. The organelles can be divided into non-membranous organelles and ones that have a membrane. Now, membranes surrounding organelles are very similar to the membrane surrounding the cell itself. They are comprised of a phospholipid bilayer, and oftentimes they are continuous with one another or in close communication with one another. So these things isolate, these membranes isolate the organelle from the surrounding cytosol. We'll see that the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, and mitochondria all have a plasma membrane. And this is especially important for the case of lysosomes and peroxisomes, because what these things contain in them are digestive enzymes. And these things you would not want loose in the cell, these digestive enzymes, because then they'd start taking things apart. So they're contained within these vesicles that have their own plasma membrane, and these vesicles will contain the enzymes in an active form until such time as they need to be used. For example, in the case of a lysosome, and we'll look at this in much greater detail later on, in the case of a lysosome, where it needs to break down a damaged organelle or an organelle that's no longer functioning. Now, we also have non-membranous organelles, and these are organelles that don't have a membrane, and they are in direct contact with the cytosol. These include the cytoskeleton, and the cytoskeleton basically is made up of your different filaments, your thin filaments, your intermediate filaments, and microtubules. And the cytoskeleton also helps give microvilli their shape. The centrioles, which we looked at briefly, which is where the center of microtubule development happens. This is where microtubules are assembled and disassembled. Cilia, which are your hair-like projections that beat in most cases. Most cilia are motile. And these are the things that allow, for example, in your bronchial tubes, things like mucus, to be brought up when you cough. And you have ribosomes and proteasomes. Now, proteasomes are interesting. These basically break down proteins, and ribosomes produce proteins. So we'll look at those in a lot greater detail as we move through the chapter. So here we can dive into the 6 types of non-membranous organelles that we just mentioned. A lot of these are structural. You can think of these all as structural. These last 2 have to do with proteins. So here we can look at some of what's important here when we look at this spotlight on page 66 and 67. You see that it's color-coded. So there's a legend up here. So our plasma membrane has this pinkish salmon-colored color on it, if you will, whereas the non-membranous organelles are contained in these boxes, or they have these boxes with these yellow, orangish labels on them. And the membranous organelles have these blue labels on them. So, for example, when we examine the centriole, which are contained within the centrosome, we see immediately that it is a non-membranous organelle. And that will help you study when you look at page 66 and 67. 66 and 67 is a very good study guide. And I suggest you look at it very carefully. Big hint, it'll probably be on the test. Certainly, it will help you remember the structure and function of all of these different organelles. So I suggest highly to study that spotlight because it tells you just about everything you need to know about the cell, at least as an overview. So here we have the cytoskeleton. The cytoskeleton is made up of these filaments, and we will see there are three types of filaments, or two types of filaments and another type of structural protein that make up the cytoskeleton. We have thin filaments and intermediate filaments. We'll look at both of those a little bit later in more detail. And we have microtubules, which are amalgamations, or I should say aggregations of globular proteins, that these globular proteins called tubulin act like little bricks and they kind of assemble together to form these long tubes. So that's sort of what a microtubule is. And then when we look at microvilli, here again, another non-membranous organelle. These are extensions of the plasma membrane. They have microfilaments in them that help give them their shape. And what do? They increase the surface area of the plasma membrane in order to facilitate absorption of materials. So we'll see these in the intestine, especially when we get into the small intestine where a lot of the nutrients are absorbed. You see these microvilli, these structures, which allow far more contact of the plasma membrane with the extracellular fluid that has these nutrients in them, and it helps bring those nutrients across the plasma membrane in much greater concentrations, or I should say in much greater amounts. Then, continuing with the non-membranous organelles, we have cilia, which aren't actually represented in the picture anywhere, but cilia are these hair-like extensions, and they have microtubules. We'll look at their structure a little bit longer. Typically, we're going to look at the motile cilia that beat. And basically, these things help move stuff across the surface. For example, when the cell faces the extracellular membrane, this is what we would call the apical surface of the cell. We'll learn what that is in just a little bit. But when you have cilia that are on the surface of the cell that's exposed to a passageway, what they do is they help move things through the passageway, as in the example of the bronchi of the where you're breathing. So air is going, basically they are the tubes that go into the lungs. So when you breathe and you get smoke or dust particles in there, you'll have mucus that contains these things. And cilia help push this mucus back up so that you can cough it out or get rid of it. There's another type of cilia that we'll see that basically acts as a flag. So it acts as a signaling kind of structure. Then we have protosomes. Protosomes break down proteins. So these things will find proteins, or I should say proteins will be sent to them that are damaged. And these damaged proteins have been tagged with like, okay, damage flag. It's called ubiquitin. We'll talk about it a little bit later on. But proteasomes will look for proteins that have this little flag that indicate they're damaged, and it's a little molecular flag. It's called ubiquitin. And we'll look, as I say, there's a slide on it a little bit later on. And so you'll know how to spell ubiquitin. It's basically says this protein is damaged and needs to be recycled. So these little proteasomes will pick up the protein and break it down into its amino acids or very small polypeptides. And then it will release the contents back into the cytosol so it can be used, those contents, the amino acids, can be reused to build other proteins. And you can think of proteo, having to do with proteins, and somes, these are little bodies, and these bodies break down proteins. And ribosomes make proteins. So this is where we have a little factory that makes proteins, if you will, and it consists of about 40% protein itself and about 60% of something of RNA. And we will look in much greater detail about how the ribosomes function and how they're made, what they're made of, I should say, a little bit later on. These things, as we've said, can be either free-floating in the cytosol, or they can be attached to the endoplasmic reticulum. Now, we can look at the cytoskeleton in a little bit more detail. We have three types of proteins that make up the cytoskeleton. The microfilaments, which are about 5 nanometers in width, they're very small, and these will form a lot of extensions. For example, when we look at the villi, the microvilli, there are microfilaments that extend up in there. And the microfilaments are often intertwined with intermediate filaments. These things are anywhere from 9 to 11 nanometers, and they will form also part of the scaffolding. Then we have these microtubules, and these aren't filaments. These are actually tubes that are made up of subunits called tubulin, which are globular proteins. And these globular proteins form or aggregate together to form these tubes. And microtubules are quite, I don't want to say volatile, but variable. And the cell can build up microtubules and take them down and build them up and take them down as it needs. These are very transient structures, or can be. So the cytoskeleton, the microfilaments are made of actin. Actin is a very important microfilament, and it's going to be quite important when we look at muscle cells. So there's another type of microfilament called thick filaments, and these are made of myosin, and these you find in muscle. You find in all muscle cells, and we'll study it later on when we get to the chapter on muscle, but actin and myosin will interact to cause movement. Typically, most other cells that aren't muscle cells don't have myosin, so we don't think of thick filaments being in the majority of cells. The cytoskeleton of most cells is made of actin, intermediate filaments, which can sometimes be like keratin, collagen, and then we can have these microtubules, which, as I say, are these tubes that are assembled from tubulin. Now, the intermediate filaments, as we say, they can be collagen. They're quite durable, and they help the cell maintain its shape, and they also help the organelles stabilize their position. So it's kind of the scaffolding on which they can stay. The microtubules are large hollow tubes, as we've said, and they're constructed of a subunit called tubulin, which, as I said, is a globular protein. When we looked at the cell earlier, we saw this thing called a centriosome, which is a body close to the nucleus, and the centriosome has two things in them called centrioles. And centrioles are arranged at 90 degrees to one another, and these make these microtubules. And the microtubules also are anchoring points, but they can change very rapidly. As I say, they're very transient structures, or can be, that can change the shape of the cell, and they also can act as a monorail system because you can move things within the cell. There are two proteins called kinesin and dynein. One moves in one direction and the other moves in the other down the microtubules, and these can help move vesicles and other cellular things that need to be transported along inside the cell. And when we look at neurons, nerve cells, we will see that microtubules are quite important in the shape of nerve cells and the axon, and kinesin and dynein are quite important for moving needed things up and down these microtubules to the distal parts of the cell. They also are important in cell division when they form something called the spindle apparatus, and this is what moves DNA that's replicated to each of the daughter cells. And you'll study that a bit later in a different chapter in second semester. All right, so here's an actual stained light micrograph where you can see in yellow here these microtubules. So this shows how they provide a scaffolding within the cell. So when we look at what some of these microfilaments do, for one, they help define the structure of the microvilli, which, as we've said, increase the surface area for absorption and attach it to the cytoskeleton. Microfilaments in the microvilli are attached to something called the terminal web, which is right underneath it. The centrioles, which are oriented, they're two very small tubes. We'll look at the structure of them in just a bit. They're two very small tubes that are made of tubular subunits, and these are where the microtubules are built from in the cell. This sort of directs the construction of the microtubule scaffolding and then the destruction of it, or I should say the rearranging of it. And they also, these centrioles are what form the spindle apparatus during cell division. Now, cilia are also little hair-like extensions that are made from microtubules, and we'll look at them as well. They have a slightly different structure from centrioles, but they too are made of microtubules. Okay, so here we have a microvillus, that is one of many microvilli. These things increase the absorption surface area for absorption. And all of these little things here are microfilaments. These are intertwined here at this thing across the cell at the what we will call the apical surface of the cell. That is the cell that is exposed to, say, a passageway or an area where there's extracellular fluid. And it has what we call a terminal web. And the microfilaments are attached to the terminal web, and the terminal web itself is made of actin and intermediate filaments. Here we have a microtubule that's helping to anchor the terminal web. And this shows you somewhat how some cells have this scaffolding that help them keep their shape. All right, here's an actual scanning electron microscope of a microvillus, or a bunch of microvilli here. And you can see the actin filaments here, the microfilaments, which are made of actin, and how they're intertwined with other actin filaments and intermediate filaments in the terminal web. Here we have the centrioles, and the centrioles are located in the centrosome. And the centrioles are made of microtubules, and they have a very particular arrangement. We have triplets of microtubules arranged in a circle, and there are 9 of these triplets, and there's no center core. So we have 9 triplets, so we call it a 9x0 array because we have 9 triplets and nothing in the center. And this is what makes up a centriole. There are 2 centrioles, and they are oriented at 90 degrees to one another, as the picture shows. And here's a cross section where you're looking in section, and you can see the 3 microtubules that make up each one of these, and how there are 9 of these total. Now, cilia also have microtubules, but they have a little bit different organization. So these can be either environmental sensors that are like little antennae sticking up, or I should say receivers sticking up that figure out what's going on outside the cell. They can be kind of signalers too, but typically they're sticking up like little radio towers, receiving information about what's going on in the extracellular fluid. And they can be used to coordinate activity around by signaling the cell to release maybe local hormones or signaling molecules. But these things basically are not motile. We also have motile cilia, and these are the ones that move. These are the ones that move things across the surface of the cell. So these have a slightly different organization than centrioles. At the base, their centrioles are very similar. Their organization is very similar to what we see in a centriole. You see 3 of these microtubules, or triplet, and there are 9 triplets. But as you get up into the motile part, the arrangement changes a little bit, and you have 9 pairs of microtubules surrounded by a pair in the center. And so this is called a 9x2 array because you have 9 of these pairs on the outside and then a pair they're surrounding within the center. And these things can move, and they have a very specific way that they move. They have what's called a power stroke, where they have, in the straight conformation, they can have a whip or oar-like action. You could think of these as like oars in a rowboat or a rowing shell, where it's moving fluid across the surface, but then it has to recover and return, and so it kind of bends until it comes back out into its fully extended. So you can think of it kind of like swimming. So when during the power stroke, say in the crawl stroke, your hand is extended and you pull it through the water, and then you bend at the elbow to return and get ready for your next power stroke. And so this is typically how cilia works. So then we can talk a little bit about the organelles in the cytoplasm that are making proteins or breaking down proteins. So we've got basically our ribosomes that we've already talked about are free and are fixed. We've talked about those, and these are assembling amino acids bit by bit, piece by piece, into polypeptides and proteins. So what happens at a ribosome, and we'll look at this in a lot more detail in the next section, is that it is receiving amino acids and it's linking them together to form very specific proteins. And the way it knows how to do this is the ribosomes are going to be receiving messages from the DNA in the center of the cell, or I should say in the nucleus of the cell. So the DNA has all of the instructions on how to make protein. We have something called RNA. We've already looked at what RNA is, but there's something that's called messenger RNA that will copy part of the DNA, copy those instructions and take it out to the ribosomes. And that will allow the ribosomes to know what amino acids to link in what order. So that's how ribosomes work as an overview. We'll look at it in more detail later in the chapter. But as we've noted before, there are ribosomes that are floating around the cytoplasm, and these things are manufacturing proteins that will remain within the cell. And then there are ribosomes that attach to the endoplasmic reticulum, and these will manufacture proteins that will either be secreted in vesicles, either within the cell or exported out of the cell. And we'll see a little bit about that first. A lot of times these things go first to the Golgi apparatus before they're even sent anywhere. Then we have the proteasomes, and these contain enzymes called proteases. An ACE is an enzyme, and these break down proteins. And these will disassemble our proteins that are no longer needed or are damaged or have been used to the point that they're past their useful lifespan. All right, so that brings us to the membranous organelles. We've already talked about what they are. We've already gone over them. But now we'll go over each in a little bit more detail. We have the endoplasmic reticulum, and this makes quite a network of flat tubular sheets. They're kind of flat, but they're tubular. They're not really flat per se, but they make this network, and a reticulum is a network. And endoplasmic means it's contained within the plasma. So and it has its own plasma membrane. So the endoplasmic reticulum, as we've said, is where things are assembled. In smooth endoplasmic reticulum, you have lipids and carbohydrates being made. In rough endoplasmic reticulum, you have proteins being made. Then you have the Golgi apparatus, which is the packaging center. And the Golgi apparatus is closely associated with the endoplasmic reticulum, because once the endoplasmic reticulum makes its products, it will modify them and store them. And we'll see a little bit about how that works later on, especially with protein synthesis. But you will have the endoplasmic reticulum making stuff and then kind of storing them for a while sometimes, and then it will send them on to the Golgi apparatus for further processing and then for final transport to their final destination. And then we have lysosomes. And lysosomes, as we will see, are made by the Golgi apparatus. Some of the proteins made in the endoplasmic reticulum are enzymes, and these enzymes can be quite destructive. And they are used to break down parts of the cell. These are like your demolition teams. These are used to break down parts of the cell that are no longer working. And demolition, for example, like old buildings, in this case, old structures that are no longer useful. So the lysosomes are contained within membrane so that their enzymes aren't functioning. And when they're needed, and you need to go demolition some old organelle that's no longer working, the lysosome will fuse with that thing and release its enzymes that will then become activated. And then in the space contained by the membrane, and not in the cytoplasm itself, but in that space contained within the membrane, the lysosome's enzymes will break down the structure that it is just fused with. We have peroxisomes. These are specific for breaking down fatty acids, and they produce hydrogen peroxide, which, as you probably know, is a free radical and can also be quite destructive to the cells. So that is why its activities are contained within a membrane so that its processes don't start breaking down other things or causing damage to other structures in the cell. In fact, it has an enzyme that will break down hydrogen peroxide into oxygen and water before it can get out into the contents of the cytoplasm. So that basically hydrogen peroxide does not get into the cell. Then we have our mitochondria. These are kind of like the refineries of the cell and the energy. Some people say they're the powerhouse of the cell because they are the energy producers. They are what produce adenosine triphosphate. So here we have our Golgi apparatus. Some say that in section it looks like stacks of dinner plates. And you can see all these little secretory vesicles coming off of it. These are vesicles that contain enzymes or protein products that are destined to be delivered either to cells outside the body or within the cell itself to do work within the cell. Here's our mitochondria, and these are the only membranous organelle that is not closely associated with all these other organelles. You can see here how closely associated with the nucleus the endoplasmic reticulum is, and the endoplasmic reticulum, the membrane, is kind of continuous with the nucleus. It's very close to it, and then it will break off these vesicles and send them to the Golgi apparatus. And so these membranous organelles are in constant contact with one another, either through membranous contact or through secretory vesicles that are going from one organelle to the other. The mitochondria is the exception. It's kind of its own thing. It doesn't have a lot of close contact with these other organelles, and it does not get membrane, or I should say vesicular products or vesicular extensions from other membranes fusing with it. That doesn't happen. Whereas the endoplasmic reticulum is going to be in constant communication with the Golgi apparatus. The mitochondria basically does its own thing and makes energy for the cell. Here we have the endoplasmic reticulum. You can see how it's sort of stacked, but you have this very highly branched tubular network. We have the rough endoplasmic reticulum, and here we have the smooth endoplasmic reticulum, characterized by the fact there are no ribosomes associated with it, whereas the rough endoplasmic reticulum does have ribosomes associated with it. So here again, the endoplasmic reticulum is the first organelle we'll look at. It has something called cisternae. Now, cisternae, you're going to see this term over and over again. It literally means a water vessel for storing water. That is where the name came from. It's a Latin word. And we will see cisternae here within the endoplasmic reticulum. We will see cisternae also within the mitochondria. It's just a term that means one of these spaces that's contained within these membranes. So cisternae, when you see this complex shape that have these flattened areas here, this is a cisterna. Cisterna is singular. Cisternae are all of these little spaces where things can go on and happen. Storage spaces. This is what is enclosed by the membrane. So they're little cisternae. So don't let that term cisterna or cisternae throw you, because you'll see it over and over again. And it's not just associated with the endoplasmic reticulum. But as we've said, the endoplasmic reticulum synthesizes proteins, carbohydrates, and lipids. It can then store some of these synthesized molecules and materials for later modification or delivery to, say, the Golgi apparatus. So it also forms kind of a network. So, something that's made over here can be transported somewhere else through here. So it also has a transport function as well, in that it can transport products from one place within the ER to another. And so we think of it kind of as a workshop, factory, warehouse, where stuff is made within it, and then it can be stored within it, and then it can be sent on somewhere for further processing. It also has another interesting function, in that it can detoxify drugs or toxins. So not only can it make stuff and store them, but toxic drugs can be brought in here and then broken down by enzymes inside. So there is another function that's not just making stuff and storing stuff, but also taking things from the outside of the cell that may not be so good. For example, things that might have gotten into the cell certain medications, for example. Eventually, I mean, you don't want to overdose on medications, obviously. Medications do have side effects. Drugs, other things, toxic chemicals you may have encountered in the environment, they can be stored and disassembled here. So the smooth endoplasmic reticulum, as we've said, has no ribosomes, synthesizes lipids and carbohydrates. So we have phospholipids, cholesterols are made here. Your steroid hormones, for example, in the reproductive system, your estrogens and testosterones will be made here. Glycerides will be stored here and made here. These are your mono, di, and triglycerides that we talked about from the last chapter. And glucose molecules will be linked together here into glycogen. And for example, we'll see this in the liver. The liver will link together a lot of glucose and the glycogen that will then be stored in muscles, and the liver has some storage of glycogen as well. Muscles can link together glycogen as well and store it. So this is where all of that happens, is in the endoplasmic reticulum. And then the glycogen can then be put into the cytoplasm as an inclusion, which is basically a non-soluble thing that can then be used. For example, in the case of glycogen, it's an energy source. And when the cell needs it, it can pick off the glucose from one by one from the glycogen. And this is a polymer where individual glucose molecules are linked together into these large chains, and then individual glucose molecules can be picked off when needed for energy. So the rough endoplasmic reticulum, as we've said, is covered with ribosomes. Ribosomes are where the proteins themselves are synthesized. Then once that they're made from the ribosome, if they go into the endoplasmic reticulum, this is where they assume their tertiary and quaternary structures. So a lot of times additional folding of the protein will happen so that it can assume its final conformation. If it wants to make a glycoprotein, it will go ahead and attach the carbohydrate part of it, so that once it has the protein, it will attach the sugar or the carbohydrate. And as we said, it folds the polypeptide protein structures into its tertiary and quaternary structures. And then it can put the product in what we call a transport vesicle. Now, the transport vesicle is like a little delivery vehicle. It will pinch off from the endoplasmic reticulum and then will travel through the cell cytoplasm on its own towards the Golgi apparatus. There's a picture of the endoplasmic reticulum. This is the rough, this is the smooth. And here we have our cisternae, where stuff goes on inside. And here we see an actual electron micrograph of it with ribosomes. This looks kind of smooth here. You don't see as many ribosomes. There are some ribosomes just floating around in the cytoplasm that are making proteins that don't need extra folding or processing, and they don't need carbohydrates attached or anything like that. They're just made and sent off freely to do their thing in the cell, whereas a lot of proteins really need that extra processing within the endoplasmic reticulum. All right, so as we say, most of the products from the endoplasmic reticulum are sent to the Golgi apparatus for further processing and for their final packaging to be delivered to their final destination. So the Golgi apparatus will receive the products from the ER through these what we call transport vesicles, and they will enter on the face that's on the same side as the endoplasmic reticulum, and they'll exit from the opposite side. Now, when we think of something on the same side, we call cis. When something's trans, it's on the opposite side. I'm not going to be too concerned that you memorize the cis and trans face of the Golgi apparatus. We'll see it in just a little bit. But just to let you know that the products from the endoplasmic reticulum comes in on the same side of the Golgi apparatus. The side that faces the endoplasmic reticulum is processed through these folds that we see that looks like in section, looks like stacked dinner plates, and then exits from the trans face, which basically faces the opposite side of the endoplasmic reticulum and faces the cell membrane. So it, as we've said, packages products from the ER. Many of these will be secretions destined for outbound travel outside the cell to other destinations, other organs, systems, tissues, and cells. These can be hormones and enzymes. and these are released through a process called exocytosis. Not only that, but the phospholipids and glycoproteins and things that we see in the plasma membrane are going to come from the Golgi apparatus. So the cell is also not just making things for export, but is also making things to maintain its own homeostasis and renew or replenish things that are getting worn out in the plasma membrane. And we also have, as we've seen in the case of the lysosomes, it will package up special enzymes within vesicles for use within the cytoplasm. So sometimes you got local delivery here. So we've got these enzymes that need to break down old and maybe damaged cellular organelles. And these will have enzymes that would be dangerous if just left in the cytoplasm on their own. And these lysosomes will then fuse with old and damaged organelles and then be digested by the enzymes within them. So you've got sort of three functions here. You can have the Golgi apparatus send things far away to other cells or other tissues. It can help rebuild the structures within the cell itself, and it can also make enzymes that will be used to demolish structures within the cell. All right, so here's a picture of it. Here's what we mean by stacked dinner plates here. They look kind of like stacked dinner plates. And these are transport vesicles coming from the endoplasmic reticulum. These were once part of the endoplasmic reticulum that pinched off and are carrying probably protein products or other products to the Golgi apparatus that will then be further modified. And then the Golgi apparatus, once they go through all these little dinner plate stack things and those processes that are going on in there are finished, like right here, it will break off these vesicles called secretory vesicles that have within them the product that are going to be used. This one is probably going to go out of the cell. The secretory vesicle here is probably going to go out of the cell and send its contents into the extracellular fluid. This one might be a lysosome, for example, that's destined for use within the cell. So here's an actual picture of one in section, you can see. And these little things also are called cisternae, no surprise there. These are secretory vesicles. You can see them how they've broken off from the main Golgi apparatus. And these are incoming transport vesicles from the endoplasmic reticulum. This is the cis face. The endoplasmic reticulum is down here somewhere. This is the trans face. That is toward the cell membrane and the exterior of the cell. All right, so now that we've talked about that, we're going to go into a little bit of protein synthesis and what happens to proteins. But before we do that, I'm going to stop here today. What I do want to show is this one picture. I want to show the process of exocytosis, because here we have a secretory vesicle breaking off from the Golgi apparatus with a product that's destined for some other cell. This is an export product. So this could be a hormone or something like that. And this little piece of the endoplasmic reticulum is going to break off with this product in it, and it's going to make its own little membranous organelle. It's not really an organelle, it's a vesicle, but it's surrounded in a pinched off piece of this membrane that was once part of the Golgi apparatus, and then it fuses with the plasma membrane of the cell, and then it opens up and shoots out its product into the extracellular fluid, and this is what it actually looks like. Here is the vesicle, the inside of it, and here it is spilling its contents out from the interior of the cell to the extracellular fluid. And so I'll stop there for today, and we'll pick back up and we'll look at some protein synthesis, and we'll look at, in a later section, we'll look at how DNA and mRNA interact, and then how the message is brought, the instruction set is brought to the ribosome, and how protein synthesis is done. So that's what we'll do next section, and I'll see you on Tuesday. Thanks.